the chance of a healthy seed being a carrier. We'll talk about it, how calculated it is for seed of being a carrier. Okay. What is the possibility of risk of being a carrier for an affected person, or brothers or sister of an affected person to be carrier for autosomal recessive disorder? We have an affected one, an affected person, okay? His brother or her brother or sister is healthy, okay, is not affected. We want to see what's the probability of this seed to be a carrier, okay? What's the probability?
50%. 50%?
50%.
Less than that?
An affected individual, okay, his or her father and mother are obligatory carriers. Okay. What's the chance of his or her healthy brother or sister to be a carrier? 100%? Why?
But obligate
100%, how do you calculate
25%? But they should be obligate carriers?
Brothers and sisters of an affected individual should be obligatory carriers. Their father and mother are obligatory carriers.
Then they'll have a lower chance, maybe.
Yeah, lower chance, but how much?
25% then. 50% here?
50% to be a carrier.
With autosomal recessive, no?
For an affected individual, father and mother are obligatory carrier. Okay? Brothers and sisters is healthy. Okay? But we want to know what's the percentage of being a carrier. Okay? Actually the chance of being a carrier is 2 out of 3. Why? Because if the parents are obligatory carrier, there is 25% of being affected for each individual, right? Affected homozygote, 25% of being healthy homozygote, and 50% of being heterozygote. Okay? But this individual is healthy. So this 25% is deleted, is omitted. He or she is healthy. Okay? There is a chance of 2 out of 3 to be a carrier. Because he or she is healthy. So we ignore the 25% of being affected. Okay? So the siblings of an affected individual, there is a chance of 75%, or not 75%, 2 out of 3 to be a carrier. Right? If we use this rule, also we will prior probability, conditional probability, and joint and posterior probability. We will find out 2 out of 3. Okay? But very simple way is to ignore the affected condition. Then you have 3 conditions. 1 is homozygote, normal. 2 is heterozygote, normal. So the chance of being a carrier is 2 out of 3, not affected. as I said, for autosomal recessive, we need to calculate the risk of being a carrier. The first thing is to find out obligatory carriers of the parents and children of an affected individual are obligatory carrier. And then each step further, the risk of being carrier will be reduced by 50%. For example, the parents of an affected individual are obligatory carrier, 100% they are carrier, each step further. Okay? This is obligatory carrier. One step further, the risk will be reduced by 50%. One step further, the chance will be reduced by 50%. One step further, this is 1 out of 2.
50 to
One
25.
step further is 1 out of 4 to be a carrier, and their siblings of an affected individual, there is a chance of 2 out of 3 to be a carrier. She is affected, he is affected. Their brother, there is a chance of 2 out of 3 to be a carrier. For an individual which is not related to this family, the risk of being carrier is population risk. Okay? It depends on how common the carrier frequency is in that population. Also, the children of an affected individual are obligatory carriers.
Carriers.
Okay. For autosomal recessive, we have Hardy-Weinberg equilibrium also. P is frequency of common allele or healthy allele in the population. Q is frequency of rare or disease allele in that population. Q to the power of 2 is abnormal homozygote or disease frequency. P to the power of 2 also is normal homozygote. And we have also combined frequency, P plus Q, frequency of common allele plus frequency of rare allele must be 1 in that population, in any population. So if you use this equilibrium, okay, P to the power of 2, Q to the plus the power of 2 plus 2PQ is 1. Okay, 2BQ is heterozygote or carrier frequency, and usually P is very close to 1. Okay, then the carrier frequency almost equal to 2Q. It means if we have allele frequency in a population which is Q, then carrier frequency in that population is 2 multiplied by Q. Now we will have some example. If we have no family history, okay, the carrier frequency in a population is 122, 1 out of 22, okay. Therefore, the chance of having an affected child for this code is 122 multiplied by 122 multiplied by 1 out of 4. Okay? You know what is 1 out of 4? If parents are carrier, the chance of having an affected child is
1/4.
1 out of 4. Why?
Because
Right? 25% chance of having an affected child for carrier parents. Okay? The frequency of being a carrier is 122 multiplied by 122 multiplied by 1 out of 4 is about 1,000. This is the risk of actually having an affected child if there is no family history. If there is a family history, okay, they have an affected child, okay, the parents are obligatory carrier, the chance of this sibling to be a carrier is, what's the chance of being a carrier for this individual? The siblings are affected. There is a chance of 2 by 3. Right? 2 by 3, and the population risk is 1 by 20. 2 by 3 multiplied by 1
by
out of 20 multiplied by 1 by 4. The chance is 1
1 by 120.
1 by 102. If the disease is rare in the population, then the chance of having an affected child will be decreased. In the previous example, the risk of population carrier frequency was 1 by 20, but in this population is 1 by 60. So the risk of having an affected child is 2 by 3 multiplied by 1 by 60 multiplied by 1 by 4 is 1 by 360. For autosomal recessive, as we said, the chance of having an affected child will be increased by consanguinity. Okay? If the marriage happened between two blood relatives without any family history of having an affected, the chance of having an affected child is will be 3 to 5%. Okay? And if the closer the relationship, the chance will be increased. So in this pedigree, we have the history of disease, family history is positive. Also we have marriage, consanguinity marriage. Okay? So the parents of obligatory carrier are the parents of an affected individual is obligatory carrier. Okay? And there is a chance of each step further, the chance of being carrier will be reduced by 50%, right? 50% this one, 50% this one. If these two individuals marry each other, the chance of having an affected child is 1 by 2 multiplied by 1 by 2 multiplied by 1 by 4 is 1 by 16. Okay. Now answer this question. What is the pattern of inheritance in this pedigree? Autosomal dominant, recessive, X-linked, unknown. What's the pattern of inheritance?
Autosomal recessive.
Autosomal recessive. Right? Why?
Because it's skipping a generation and both, for example, in generation third, the fourth and the sixth individual are the affected people, while we don't see any individual in generation one and two have the disease.
Because affected individual has normal parents.
Yes,
Okay?
exactly.
Normal parents have affected children. Right? What is the risk of having an affected child if 3-1 married with 3-4? 3-1 married with 3-4. 1 by 2, 1 by 4, 1 by 8, 1 by 25. Three one, 3-4. The first thing that you need to do is to find out obligatory candidates, right?
There is an obligatory carrier. Yes, one of the parent will be an
Which
obligatory.
one?
3, 4.
3-4 is affected.
I keep confusing between these.
Carriers. We need to find out carriers.
Okay.
In
We
second
know.
generation, fourth and fifth may be the carriers. In second generation,
Second generation,
fourth and fifth are the carriers probably.
yes. 2, 4, 2, 5.
Exactly.
They are obligatory carriers,
Right.
right?
Yes.
Because they have
Affected offspring.
affected
Offspring.
children. Okay? So, one step further, the chance will be reduced by
50%.
50%, right? So, the chance of 2-1 to be a carrier is
3.5, 2.5.
this one is obligatory carrier, this one is
1/8.
50% chance, 1 out of 2,
1/8.
okay? 50% chance of being a carrier. This is 100%, one step further, 50%, one step further, 1 by 4. There is a chance of 1 by 4 this individual to be a carrier, right? This one is affected, he will transmit the gene, the mutation to his children. All of his children will be get the mutated allele. So the chance of having an affected individual if 3-1 married with 3-4 is 1 out of 8. Why?
Because of the
100% this individual to be a carrier, right? 50% this individual to be a carrier, 25% this individual to be a carrier, right? And this 25% multiplied by
half. Again,
50%,
50%.
right? 1 out of 4 multiplied by 1 out of
2.
2. Because
1 out of
if
8.
she is a carrier,
There's
there is
50%
of
chance.
50% to transmit the allele to her children. Okay?
In the second generation, because as like In 2-4, they are like sure obligate carriers, right? 2-4.
2,
4.
4. She's obligated.
Yes. So we're going like 2 steps away when we go to 2-1, right?
Not 2 steps, just 1 step.
1 step.
Yes. Sisters of this one.
That one.
This one.
No, on that side.
This one.
Yes, that's
Not, this is not 1 step, 2 step, 3 step.
Uh-huh.
This is just 1 step.
Okay.
All of them, all of these 3 individuals are 1 step further.
Okay.
1 step, they are brothers and sisters of this individual.
Okay, in that sense.
Oh,
Okay.
okay.
Yeah.
All these 3 individuals, there is a chance of 50% to be a candidate.
Mm-hmm.
All these 3 individuals, they are 1 step further, right?
Okay.
And 1 step further, from
Downwards.
2 to 3, from 2-1 to
3-1.
3-1. So 1
1
by 2 multiplied by 1 by 2
2.
is 1 by
1 by 4.
4. If she is a carrier by 1 out of 4, then there is a chance of 1 out of 2 to transmit. So the chance of having an
1 by 8.
affection is 1 by 8, because this individual is affected and he will
I have a 50%
transmit
chance.
the gene.
Mhm.
Okay. What is the risk for couple 2-3, 2-2 and 2-3 of
3-2
having an
and
affected
3-3.
2-2, sorry, 3-2,
3-2.
3-3. It's 2 individual. 3-2 and 3-3.
Sorry, 5.
1 by 2, 1 by 4, 1 by 8, 1 by 24.
1 by 4 and 1 by. Again, 50% was right. Still going to be right? No. एक,
1 by 8.
1, 4.
How? Why?
Because there it has to be 1 by 4, 3, 2, and
1 by 4, 3, 2. Right.
for 3, 3 it should be 1 by 2.
Why by 1?
Because it's 1 step away from the sibling which is affected. No, in third generation.
This will be 3 by 2 by 3.
1, which one?
In
There is
third
a-
generation.
2 by 3 into
in this
1
one
by
exactly,
4
1 by 4.
equals at 12.
Which one in third
In
generation?
second.
Give me an exact address. Give me an exact address. 3,
3 and the second.
3, 1, 3, 2, 3. Which one?
3, 2.
3, 2. This is exact address. Okay? 3, 2.
Yes.
1 by 4. Right. To be a carrier.
Carrier. Yeah.
So for this one,
It's 1 by 2.
1 by 2.
Yes.
It's 3 by 2? Oh, 2 by 3?
2 by
Sibling,
3.
sibling of a
2 out of
sibling of a
3.
carrier.
Why?
Sibling of a affected person.
Siblings of an affected individual, there is a chance of 2 out of 3 to be a carrier. Right?
Mhm.
So 1 by 4 multiplied by
1 by 2 by
2
3.
by 3, then multiplied by
Now they'll multiply by 1 by 4.
1 by 4. Right. If these 2 individuals are carrier, there is a chance of 1 by 4 to transmit, both of them to transmit their
48 half. 1 by 24.
alert to their children.
1 by 24 is the answer.
1 by
24 is the
24. Right? Right. Any questions? Okay. Problems for embryologists and genetics they had some question about human development. We have more than 10 to the power of 12 cells in our body. Each cell has a specific appearance, a specific function. We have muscle cells, we have bone cells, we have eye cells, we have liver cells. They have different function and they have different actually appearance. And there was a big question of how these different cells comes from one fertilized egg. We said our genetic information is stored on DNA. Okay? The DNA of these all cells are the same. Okay? The same DNA we have in our liver cell, the same DNA we have in our skin cell, in our muscle cell. DNA is the same, but the function of these cells are different. They have the same genotype, but they have different phenotype. Okay, how this happen?
Variation.
Without changing DNA, the function of cell is changed. Without changing the DNA, the appearance of the cell is changed. Okay. Some genes are expressed in our liver cell. Some genes are expressed in our muscle cells. The genes that are expressed in our muscle cells, maybe they are not expressed in our liver cell, or the cells that are expressed in our liver cells, they are not expressed in our muscle cells. How this happens? The DNA is not changed. Okay? The same DNA we have in all cells. The expression of gene is changed based on epigenetics. Okay? Epigenetic is the actually mechanism of differentiation of the cells. Cells differentiate from one fertilized egg. One cell become liver cell, the other cell become muscle cell, the other cell become heart cell, bone cells, skin cells. All the same DNA, but based on different expression, they differentiated to different cells. Okay? That was the questions, the big question for the embryologists, and then they actually introduced the word of epigenetic. It means on the top of the genetic. It means something more than the genetics. Genetic, classic genetic. Also, there was another question for embryologists about the diversity of phenotype within a population. We all have the same DNA. 99%, 99.9% of our DNA is the same. Human, from Iran, from Iraq, from Pakistan, from US, from every country, about 99% of our DNA is the same. But diversity between the human is more than 1% different in our DNA. Okay? And also another question was about twins, monozygote and colon animal. Monozygote twins has the same, 100% the same DNA. Colon animal has 100% the same DNA. But there are some differences between them. How this happened? Because of the changes in expression of the gene without changing the sequence of the DNA, without changing the structure of DNA. The expression of gene is changed based on the epigenetic mechanism. Also, the relationship between the phenotype and the genotype. one individual has mutation in the one gene, and the phenotype will be different from another individual with the same mutation in the same gene. The relationship between phenotype and genotype also are different based on the epigenetic mechanism. The meaning of the epigenetic mechanism actually is changing the expression of the gene without the changing of the DNA structure, DNA sequence. You know about this slide? I show this slide in any session to remember your single gene, chromosomal and multifactorial disease. And we also had this slide in molecular genetics. We talked about the regulatory region. What we are going to talk today usually is related to regulatory region, not coding region of the DNA. We said regulatory region is a region that regulates the expression of the gene. Okay? The mechanism of epigenetic usually have any regulatory region that causes the actually changes of the expression in the gene. Usually epigenetic involves in many mechanisms, in different mechanisms in our body, in cellular differentiation and development, in metabolism, in some diseases, phenotype variability, in inheritance, evolution, behavior, even in our culture. The changes in epigenetic can cause the difference between our cultures also. Many diseases develop when the wrong type of epigenetic or epigenetic introduced at the wrong place or wrong time. What does this sentence mean?
Epigenetic mix.
What was the meaning of the epigenetic changes in the expression of the gene without changing in the DNA structure or DNA sequences? Okay? If we have wrong epigenetic, it means wrong expression of genes happen in wrong place. What does it mean? And wrong time.
Like some of the example methylation has occurred
What?
in excess or in less methylation, like addition of methane or something like that.
Yeah. What does it mean by right place?
Where it's not supposed to go.
Place is our organ, our tissues. Okay.
Okay.
Some genes, for example, must be expressed in our muscle. If they are not expressed in our muscles without changing the DNA, okay, DNA sequence, the gene is not expressed in our muscles, okay, wrong place. That gene goes to express, for example, in our bone, wrong place, okay. The expression of gene is changed, the protein is not made, the muscles cannot do their function, then we will have some genetic disease, some disorders, not genetic disease. Okay? Those genes that must be expressed in our muscles is not expressed, and then it's expressed in our bone, for example. Or those genes that must be expressed in our liver is not expressed in our liver, it's going to be expressed in wrong place, in other tissue, for example, in our stomach or in other places. So we will have wrong epigenetic in wrong place. So what about the wrong time?
Maybe it's happening at fetal period or like in different
It's like about the age.
age. Maybe it's related to age.
Age. An example.
Like in maybe gigantism, there is from the beginning, like
Some genes, for example, must be expressed during our fetus age.
Yes.
For example, hemoglobin. We have fetus hemoglobin, which is expressed during our embryology time. Okay. If these after the birth, these genes must not be expressed. Okay. If fetus hemoglobin is expressed in adult, wrong time. Okay. Then we will have some disorders. Okay. If it's not expressed in adult, if it's continued to be expressed in adult, we will have some problems. Okay. So wrong epigenetic in wrong place or wrong time, we will have some epigenetic diseases. Okay. Without changing of DNA sequence, without any deletion, without any mutation, without any problems in genetic structure. So this is the genetic, epigenetic actually disease. So in parallel with epigenetic term, we also have other terms which almost equal to genetic. We said genetic. Besides the genetic, we said epigenetic. We talk about genotype, we can talk about epigenotype. We talk about genomics, we can talk about epigenomics. In parallel with the genetic, we have other words that is almost similar. For example, when we say epigenotype, it means epigenetic state at a locus. Okay? When we say about epigenomic, it means a study of epigenetic on the genome-wide scale. Okay. What does this slide show?
The methylation in between the the M in the third part explains the methylation.
There is a more meaning of that methylation in this slide.
More than that. Font of DNA sequence. The way it's written as well. The font, the style of the basically letters and sequence of letters. That's what's written.
Yeah.
So how would that?
We said epigenetic is the changes in the expression of the genes without changing in the DNA sequences. Okay. We have 4 letters here, right? C, A, G, T. They have been written in different fonts, different size, different colors. Okay? The letters have not been changed. Letters is the same. But how you can read it is different. Okay? Maybe you can read one font easier than the other font. Okay? The expression of the gene is more. Okay? The reading of this letter is different. It's something like this. Just changing the font, changing the color, changing the size, without changing the letter, without changing the DNA sequence. The expression will be different. Epigenetic is something like this. Changing the font, changing the letter, changing the color, changing the size, without changing the sequence. Without changing the letters. The appearance of these, for example, maybe you can read this one easier. You can read this one easier than this one, right? Or this one is harder to be read than this one. Okay, the expression is different without changing the sequence. So by this actually definition, then we can change the categorization of the diseases. Okay. We said we have single gene disorder, chromosomal disorder, multifactorial disorder. But in addition to them, we can have epigenetic disease. We can divide the disorders to genetic disease. Genetic disease are the disorders that happen because of the changes in the single gene, because of the changes in the chromosomes, because of the changes in the several genes, gene and environment, change our genetic condition, and then we have genetic disease. We also can have epigenetic diseases. Without changing the DNA sequence, without changing the gene, without changing the chromosome, we will have some diseases. We call them as epigenetic. And another category is actually mixture of both. Both changes happen in the epigenetic and genetic. So then we will have some diseases. So epigenetic is the mechanism of changing the expression of the gene without changing the structure of the DNA. There are three mechanisms for epigenetics. One is methylation of the DNA,